Hello, everyone. Welcome to the November Caribbean Connections Team Meeting, or CCT meeting. I'm AGRRA's Judy Lang. Our program director, Patricia Kramer, is at the Zoom controls. Today, we are very pleased that we also have NOAA's Dana Wucinich-Mendez, who in 2018 founded the CCT after stony coral tissue loss disease, or SCTLD, reached the Caribbean. Please note that closed caption translation features are available. Please say hello in the chat by writing your name, affiliation, and country, and email if we don't have it already on our records. This meeting is being recorded. The recording will be posted on the AGRRA webinars page. Once available, you will be notified via email, providing we have your email address. As many know, this year we have been promoting coral rescue programs with our partners in the hopes of collectively preserving some genotypes that are representative of our region's unique endemic biodiversity for those corals most at risk of local ecological extinction due to SCTLD, which is a biotic disease. However, the catastrophic heat waves of 2023, which extended across the entire northern hemisphere, are a signal that we can't ignore the existential vulnerability of corals to the dangers of warming from seawater temperature incursions. Bleaching is considered an abiotic disease in the medical and veterinarian sciences. And what does that mean? Well, today we are fortunate that we will begin with a presentation by our marine invertebrate histopathologist and environmental scientist, Dr. Esther Peters of George Mason University, who will introduce us to the new understanding of what is meant by disease. You are encouraged to write your questions in the chat, and we'll have some time for discussion after your talk. So Esther, please take it away.
Thank you, Judy. Started. Okay. Hello, everyone. Oops, we can start in the right place. Okay. Hello, everyone. Judy continues to ask me questions about coral diseases, and she decided that you might also like to know my replies. So here I am. Due to limited time, I will not be able to go into detail about everything in this PowerPoint, but you can download the PDF through the link that I posted in the chat, the very first one, and send me questions later. First, I want to thank everyone for their interest in coral diseases and their contributions. As Terry Work said, we have lots to do, and we need lots of people working on them. Beginning with what is disease, because I'm not sure everybody understands this. It is any impairment that interferes with or modifies the performance of normal structure and functions. It can include responses to environmental factors, the abiotic factors that we talk about, as well as infectious microscopic organisms. So what we also know is that health is the state of an organism when it functions optimally without evidence of disease or abnormality. These are very important definitions to know. It's not moving. Yikes. There we go. Criteria for disease. And let me get this out of the way. At least two of the following, not all three. You could have a recognized etiologic agent, and remember, a pathogen is a pathology generator. So it's any virus, microorganisms, or other substance that can cause disease. There's an identifiable group of signs, and in the case of humans only, symptoms, and there will be consistent anatomical alterations. we know that our molecules that form our bodies and the whole world, of course, but that they are have bonds among them, and we know that those form the structures that we see that allow the functioning of our bodies. We have an example of this in Alzheimer's disease, where we have signs, symptoms, and brain changes, but we don't know the pathogens yet. So we know that for coral diseases, this are the criteria that we have set, that when they appear to be healthy, and we have learned that functional impairment is found when we have abnormal behavior, unusual tissue coloration, abnormal skeletal deposition pattern or rate of deposition, a growth anomaly, or slow or rapid disappearance of tissue from the skeleton. But what we also need to remember is that the healthy coral, apparently healthy coral, may not be that, because at the molecular level, molecules, the bonds among them, and what functions they can perform may already be changing after exposure to biotic or abiotic pathogens. That much more cell and tissue damage usually has to occur before we can visually detect lesions way out here in the duration of the injury. And sections are shown here from 2 Orbicella colonies that were both apparently healthy. The upper one shows the basal body wall within normal limits with abundant lipid droplets in it. The lower one is showing the basal body wall full of degraded zooxanthellae. We don't know why that happened. As diseases have been studied in plants, animals, and humans, various paradigms or models of disease were proposed. The first was Robert Koch's in 1884, 4 criteria. Single infectious agent causes one disease. And there were other things that you had to meet to identify that. People have found that there can be asymptomatic cases, microorganisms that cannot be cultured, and other issues. So they began to wonder what was going on with all of that. In 1965, Bradford Hill came up with his rules of causation to help define when associations could be interpreted to be causal in epidemiologic studies. And he thought that there would only, again, be one single factor as the cause of the disease. But that was not necessarily going to work. We have this long list now of potential pathogens. It includes both abiotic and biotic factors. And the epidemiologic triad that I show here in the middle is what explains disease. We have a model in which we have the host, the pathogen, which is a subset of the environment and influenced by the environment, and the environment that is where the host and pathogen can intersect and potentially disease can occur. And the environment also affects the host as well. We talk about an optimum envelope in which the levels of all of these potential pathogens are at ideal concentrations, let's say, or intensities. And if you continue the exposure outside of this envelope to too high or too low levels, then the host, then what the host is adapted to, and homeostasis cannot be maintained, then disease or death or results. The latest paradigm builds on all of these findings to include predisposing factors. What the organism has been exposed to, every exposure to which it has been subjected from conception to death. This was brought out by Wild in 2012. So this is a model that has been drawn. We have general and specific external environments. We have an internal environment that is driven by what our genes allow to happen there in terms of our proteins and enzymes. And but now we look back at this over all the lifetime, and we know that it is dynamic, that all of this is constantly changing. How can we wrap our heads around this when we look at corals? I'm going to talk a little bit about insights from tissue loss and tissue discolorations with the focus on Acropora today. Note that we usually have to do a lot of observations to compare and contrast similar disease signs and other field and laboratory results to determine which of two or more diseases is present. The same signs could have different etiologic agents or the same, and sometimes the coral individual or species differences may affect how we can interpret different diseases. So you've got to study multiple parameters. You've all been familiar with the many phases of tissue loss in the acroporids, and there are even other morphological changes that have been reported. But what do we know about etiologies? Well, things could be different. I first found in 1983 bacterial aggregates, but then others didn't find them, including me. And what did that mean? In the 2001 in white pox, Serratia marcescens, the enteric bacterium was identified as the pathogen, but it has not been seen since 2013. And of course, we have Vibrios, we have other bacteria, we have ciliates. A lot of different things could be causing the tissue loss. In the Florida Keys in 2003, we sampled corals, acroporids that were losing tissues. And in White Shoal, in the dry tortugas, we found examples where the coral might have been affected with Serratia. And then we also found them with the ovoid basophilic bodies. So with all this confusion going on, the Forest Rowers Lab published a paper saying that they had studied Acropora cervicornis in Panama, and they had this widespread association of a Rickettsia-like bacterium, or RLO, as we say, that could not possibly be the pathogen because it was present in both healthy and diseased staghorn corals. But those RLOs are obligate intracellular parasites and known pathogens in other animals. So we're working along with others. We identified them using a special stain. You can see them on the tentacles and oral disc in a coral, on the nidoglandular bands of the mesenterial filaments, and in the mucocytes of the epidermis. They were only infecting the mucocytes. And this has now been identified as Aquaricatia roweri. 100% of the Caribbean acroporids that I have examined histologically since the 1970s have these. They are infected. And we now know that we have present populations of acroporids in weakened condition. So we have this chronic disease there. We also know, finally, that the diseased coral may not be having this disease only where the skeleton is showing, that the surface epidermis, where the zooxanthellae are present, may hang on for a long period of time, as you can see in this photo on the right, even though the rest of the coral underneath it has been dying. Moving on to bleaching. Well, this is an important sign of disease, of course, and what it means when we lose the endosymbiotic algae or their pigments. There's a metabolic imbalance occurring in the tropical corals, and that is a functional impairment. So it is also a disease. Abiotic factors, of course, were first identified: temperature, even changes in salinity, light, UV radiation, and combinations of factors. But these are some examples of different bleaching situations under the microscope. In the upper right, we have a bleached coral in which the zooxanthellae were not expelled or exocytosed. They're still there, but they're misshapen and shrunken. They're not obviously producing pigments anymore, and the coral cells are beginning to lyse. In the middle, on the left, we have a coral where the zooxanthellae were exocytosed, and it looks in pretty good shape, so maybe it can recover when conditions get back to normal or other things happen. And then we have on the right in the middle, the loss of algal symbionts, but you can see there's also been tremendous damage to the tissues. The cells are vacuolated and there's just a loss of them. And then in the lower right, we have a case where the gastrodermis was completely sloughing off the coral. That's not ever going to recover. And in the lower right, we have bacteria, and that may not ever recover either. We now have identified all of these biotic agents of bleaching, including the Vibrios. We have corallicolids. They are apicomplexan or protozoan parasites that get into gastrodermal cells and replace the zooxanthellae. We have viruses that infect and kill the endosymbionts. And we even have an unknown cause that I still haven't reported on because I don't know what to call it. Something came in and changed biochemically the collagen in that coral. So we get back to our big web of causation, lots of different things going on that lead to stress and ultimately disease. I made my own model of this for our tissue loss in the acroporids. If you have the proper levels of the various environmental conditions, then you're going to have corals that will grow and survive and reproduce. But if we get changes that lead us to high levels of stress, high levels of these things like temperature or pH, or we get low levels, those are going to lead to stress, and those are going to change the way the coral is able to function. But as you can see, it can go from one to another with this multifactorial thing going on, this chronic infection as well as susceptibility to other pathogens in the environment as well as biotic ones. And of course, overall, we have, unfortunately, the human population increases, the elevated CO2, the temperature elevations that began, oddly enough, down here in the 1970s and have continued rising. And we now have ecosystem distress syndrome, as people are saying, that is overarching our concerns. Two brief notes: the One Health Triad, not about zoonotic diseases or infectious pathogens, but also about ecosystems. We know that we have to have, in order to have healthy people, we need healthy animals, we need a healthy environment. And I wanted to say, if it is not safe for corals, it is not safe for people or other organisms. And so we need to get that message across. And a couple of important reminders from pathology, the study of disease that has been going on for millennia. The disease names are not capitalized and disease is not transmitted. Only infectious pathogens that may cause disease can be transmitted from one host to another. So with that, I will end and be happy to answer any questions.
Esther, thank you very much. And we do have time for a few questions. And maybe I would like to start by asking you, okay, how can we refer to coral diseases if we can't say that SCTLD infects them? With humans, we talk about catching the flu, or people get TB or get tuberculosis, or they have COVID. What words do you wish us to start using when we're referring to what we see in the field and give a field name for a disease, or we don't know what we're looking at?
We can simply say it is being affected by something that is causing what appears to be a particular disease.
That's a very long phrase. You can't just say
it's a very long phrase.
You can't just say it has. We think it has SCTLD. It might have.
You could say we think it has this disease, but that doesn't say anything about the causal agent necessarily, because Absolutely. We're still trying to figure it all out.
Yes, exactly.
Yeah.
So we think it has SCTLD as long as we understand that we're only talking about the signs that we see underwater.
Right. That's correct.
Okay, good.
Thank you, Judy. Real quickly, Esther, could you go back one slide? We had a request for you to go back to that one. And we do have some questions from our group here today. I think, Daniel, you had your hand raised first, and then we'll work our way around the room.
Yeah, thanks. That was really excellent overview. I learned a lot. Thank you. I was wondering if you could refer back to the slide that had the pathology screens. I wasn't sure really, to be honest, what I was looking at. Yeah, here, perfect.
Mhm.
I was wondering if you could like just help orientate me to these a little bit, like where are the symbiodinium within
Oh.
these slides?
Yes, so in this normal slide, here you see these nice little round, circular, spherical, actually in 3D cells of the symbiodinium.
Okay.
Or
Yeah, and
symbiote.
that's one of the tissue layers of the coral, right? Is that
Right,
like the
right. And here you see them here, but they're separating as the gastrodermal cells are lysing. But in here we don't have any. But here they are there, but they are not nice and round anymore. And this coral was said to be completely bleached, and yet those cells were still there, but they had died. They were not producing pigment anymore.
Yeah, that's really fascinating. So it sounds like the model we usually hear where the symbiodinium are simply expelled. It's more complex than that.
It is more complex than that. Thank you for getting that.
Thank you.
Okay, thank you very much, Daniel. Esther had a quick question from Lisa. She had asked if you could please explain role of vectors as well.
Oh, yes. So a vector is an organism that can take up an infectious microorganism and move it from one host to another in some way. And of course, we think of mosquitoes, mosquito-borne diseases, viruses, bacteria being moved around from one host to another. But in the sea, we probably also have vectors, and these may be fishes that feed on corals. I had a student who tried to look at the 3-spot damselfish as a vector for the tissue loss in staghorn corals. Didn't really find if it might be doing anything there. But we also have, tons of little things such as copepods that could be picking these things up. Remember, copepods picked up Vibrio cholerae, and that is how cholera gets spread around. But if you filter your water through 6 layers of silk, you can get rid of those and you don't have cholera anymore. So there's and a lot of things, and of course the corals do feed on those, and we've got to look at what may be coming in, how they may be getting them from different sources.
Thank you, Esther. We have time for 2 more questions. We have a question from Maya, and then we'll go to Andrew. And you can continue to put your questions in the chat, and we will follow up with questions, especially if we have more time at the end. But then also Esther will take time to reply to all questions that are asked. So Maya, if you want to go ahead and ask your question.
Thank you, Esther, for a great presentation. I love the one ecosystem health slide and concept, and curious to know what's needed to bring that discussion to communities in collaboration with people who've worked on COVID, people who are working on dengue right now, as many coastal areas are grappling or have grappled recently with these things. What do we need to accelerate that type of convergence?
Well, we need to keep talking about it. We need to know that there is an international organization for One Health. You can get One Health newsletters. This is a huge thing for veterinary and medical people, as well as plant pathologists. So it is out there, and if you want, I can send you other information.
Thank you, Esther. Thank you, Maya. Andrew, would you like to ask your question? Oh, go ahead. First, Judy, you can reply, and then we'll go to Andrew.
Yeah, real quick before Andrew. There is a chapter of the One Health group in Jamaica, and they have meetings once every year and bring together veterinarians and medical practitioners of all types, from anatomical types to psychologists, and the meetings are also being held now virtually and are open to the public. So if anyone is interested in finding out what's happening in at least one part of the Caribbean, I can help you afterwards. Okay, thank you.
Andrew.
Need to turn on myself, don't I?
You're good.
Yeah, here we go. Good morning, everybody. Esther, I was just wondering, back to the same slide that you had for the first fellow Victor, I want to say, with those differences in the reaction, basically in how the bleaching is happening, are there morphological differences? We just saw through this bleaching event, we've seen fantastic plasticity differences in how heat impacts the different individual corals. Some of them just turn uniformly white all over them. Some turn uniformly white with retaining some color underneath, and some just get into this weird patchwork of browns and whites. And it was but almost like a sort of a non-uniform fade. So I was just wondering if those reactions that were in your slide, if we can see them in the actual animal.
Well, oh yes, because those came from actual animals. Unfortunately, not all of them were accompanied by underwater photographs. So I don't know, and more work needs to be done on that, certainly. But we, I've seen all those variations myself, and I've seen a variety of things under the microscope. So, and this could be due to, of course, the individual colony and its genotype, and whether it is able to moderate the influence of the pathogens that may be affecting it and its responses. And it also looks at the level of the length of time to which an organism is exposed. That is also very important. Dose plus exposure levels. So there are going to be variations.
Okay.
Yeah.
And then and then here and there we were finding one. I think Felix is going to talk about them. That just had no reaction at all, which was just really something.
Yeah. Definitely.
Hello, I have a question.
Can you put that
Yep.
in the chat, please?
Okay.
Because
Yeah, we're gonna, we need to move on to the next set of speakers, and at the end, if we have more time, we will have more questions and discussion. So we will have another question and discussion period at the end of the next set of talks. And in the meantime, I would encourage everyone, if you do have questions, please write them in the chat. And Judy, back to you, so you can start the next set of examples of mortality, resistance, or rescue.
thank you, Patricia. The high seawater temperatures that were devastating corals in the Western Caribbean and Florida during our September meeting have now virtually filled the entire Caribbean basin. Temperatures are still above 30 degrees Celsius in some areas. Bleaching is still ongoing, and final assessments are premature. We will have 3 talks exemplifying what is happening, mostly to the particularly vulnerable species of Acropora, and then hear about a huge rescue of mostly SETLD susceptible corals in Florida. You are, again, I'll say it again, encouraged to write your questions in the chat, and there will be time to talk with all the presenters after the 4th talk. And first up are Genevieve Renaud-Byrne of the Elkhorn Marine Conservancy and Ruleo Camacho of the Antigua and Barbuda National Parks in the northeastern Caribbean, who will describe RTL disease and bleaching in Antigua. So please.
Thank you, Judy, for such a great intro. And also just want to say thank you, Dr. Esther, for such a great talk. Really, it was quite eye-opening. All right, so what myself and Jen are going to present today is just kind of an overview of what we've been seeing over the past couple of years, particularly over the summer. And what I want to start out first is looking at sea temperature. So I've been recording sea temperature since June last year using Hubble temperature loggers. This log is mounted at around 25 feet deep in on one of the nursery trees, and it's recording a temperature every 10 minutes. And particularly in this year, we really started to see problems with RTL around April, May, where you see that first jump. And that first jump in temperature is where we started to see major RTL issues popping up in the nursery and an occasional bleaching. And we were following along with what was happening along Florida's Panhandle and what's not at that point. But then late August, we had a second jump in temperature that really spelt doomsday for the corals around Antigua and Barbuda. We saw mass bleaching throughout almost all species, which you'll see going forward. Jen?
Yeah, thanks, Ruleo. And just as Ruleo mentioned, in that April, May period of time where the temperature spiked, we started seeing rapid tissue loss, or corals being affected by an infectious organism or pathogen. We're not really sure exactly, but we identified it as RTL because of this tissue sloughing off the skeleton, as you can see in that picture there on the right. We started monitoring this in our nurseries just so that we can make better internal decisions as to which corals to propagate, and to see which genotypes were less susceptible. According to our observations, susceptibility seemed very genotype specific. We observed higher susceptibility in ACER genotypes, followed by APRO and APAL, as you can see on this graph here. And it's really unclear if proximity to other infected structures or location within the nursery played a role for us. I don't know, Ruleo, if you want to add anything about RTL in your nursery.
No, please continue.
Okay, well, that was it. We can go to the next slide. Yeah, and just as Ruleo mentioned, in that late August period, we started witnessing mass bleaching all over Antigua. This is a picture of Hell's Gate within the Northeast Marine Management Area. You can see here a picture in 2021 that we took of the exact same site. It's probably not the exact same slope, but this huge field of APRO was pretty much 100% bleached. A lot of it had already died, which is why you can see some of it is white and some of it is looking a little bit darker. That's because cyanobacteria has started to attach the skeleton that has deceased, unfortunately. Next slide, Ruleo.
Jumping back in here, what we started to do was try and, as we're seeing this bleaching, we noticed that it was happening on large scales throughout the nursery. And so what we did is, doing assessments. We did an assessment in September, and by mid-September, we had already gone down to about 7% health in the nursery. What I will say here is healthy corals could be corals that may have suffered from RTL in the past, but had healthy living tissue remaining where you could clearly see an intersection where the disease progression had stopped, and you could see fresh growth starting over. Whereas anything that was bleach or pale or dead missing was things that we had trimmed off over time. So we had 7% healthy corals in September. I think we did another assessment in October that was down to like around 6%. But we've just seen this massive sweep through both nursery systems and parent colonies that has been very concerning. We've uploaded quite a bit of data to the AGRRA's bleaching website. But the sweep across colonies, it kind of got to the point where we were doing what we can to try and ensure that the nursery systems were surviving as best as they could by removing excess dead coral structures, cutting off areas where we're seeing bleaching. And then the other thing that we did notice is once you had that temperature spike, we had a complete elimination of RTL. So whereas we had RTL leading up between May to August, and you'd see increasing amounts of RTL. And I remember in the last talk, that was something that came up where we're convinced that we theorized that the RTL was actually being driven by this elevated temperature. Once the temperature got to a certain point where we started to see bleaching, the RTL more or less disappeared out of the systems, and we're just seeing bleach corals.
Yeah, exactly. And And just to kind of follow up on what Ruleo just said, I'm not 100% sure if the RTL has stopped or if we just stopped seeing it because the whole coral went white. But anyways, we noticed bleaching in our nurseries in mid-September. We started collecting data on September 25th. I've just put in some data here for you all to look at. On October 9th, nearly 3/4 of our nursery was bleached, but we had minimal mortality, so it just kind of had started. And then only 1 month later, mortality climbed to just over 60%, which was obviously very difficult for us to watch. And 15% of those corals were still thermally stressed. Now, the good news is that the number of healthy corals has lowered, has climbed in that 1 month period from 16% to 22.6%. So that's some good news there. And that is across 62 genotypes and just over 2,400 coral fragments. Something that Judy was interested in knowing more about was the temperature in the day versus at night. We looked at the data and we noticed that the change in temperature was about 0.5 degrees Celsius. The max decline at night that we saw was 0.8 degrees Celsius. So it's quite significant. And it seems to be that the coolest temperatures are at the very end of the night. And right now our coral nurseries are back down to 29 degrees Celsius. So we hope that our survivorship will continue to increase and that thermal stress will decrease over time. Next slide, Ruleo.
What I'll just add there to what Jenna said also is, and this is a question that we've gotten from Judy also, is whether or not we're seeing any stratification in terms of bleaching. So whether corals at the top of a nursery tree seem to be more bleached than corals at the bottom, which would have indicated that maybe shading could have been a potential solution. And that is what we are seeing, is that throughout the nursery trees, we're seeing bleaching essentially at the same rate. Whether at the bottom of a nursery tree, which in the NPA's nursery is at around 28 feet, or at the top of the nursery tree, which is at around 18 feet, right? Because these are 10-foot structures. So we were seeing uniform bleaching throughout, which was indicating that there's no necessary preference in shading. And we were getting reports from dye shops and other areas that similar temperatures were being seen on a regular down to 60, 70 foot deep. So this warming temperature is actually something that was going through throughout. Just to jump in here, in terms of wild species, this is a running list, I would say, that me and Jen have been working on in terms of what we've been seeing bleached. And this list includes corals both in Antigua and Barbuda. So far, I haven't observed a species that hasn't been affected. I will say that the thing that seems to be least affected from what we're seeing is probably MCAV and SIDS. They seem to be, at least from what I've seen so far, those seem to be the least affected corals. But everything else, every type that we've seen, including the fire coral group, has appeared or has signs of bleaching or been completely bleached in the field.
Yeah, I have nothing to add there, Ruleo.
All right. So this slide here is kind of a ending point and kind of a combination of images from Jen and myself. Because one of the concerns that we've had, and I mentioned earlier, is that a lot of the parent colonies that we were using for nursery propagation have been bleaching in the field. So there's been a lot of work trying to find and identify colonies in the wild that are doing better. And this is work, whether scouting by the dive shops or scouting by us within nursery systems. And we have seen some hope. The concern that I have is a lot of the stressor hasn't completely removed itself. So we're still seeing these elevated temperatures while these corals seem to be surviving. It's whether or not the stressor will remove itself in time for them to be considered viable alternatives to repopulate nursery systems, or if the numbers will even be sufficient for repopulation.
Yeah, and I think Just to note that we've done swims both in Cades Bay Reef, which is on the western side of Antigua, and then throughout the Northeast Marine Management Area, which really extends the whole northeast of the island. And we've seen many, many colonies of APALs that have seemed to be unaffected, which is really great news. They are few and far between, so we're going to have to help them out. But there is signs of hope, and we look forward to doing another swim in a couple weeks' time when hopefully the temperatures have returned to a normal or a cooler temperature, and we can see what's left. That's all I have, really.
Thank you very
That's
much,
close.
Jen and Ruleo, and also for anticipating some of the measures that you may undertake in the future. We will next hear from Lisa Carne of the Fragments of Hope in Belize, who will address what's happening in the far western Caribbean, where the temperatures in excess of 30 degrees were already achieved last June. And so the stressors has been applied for an extremely long time there. Lisa.
You guys have to open up my video. I can't do it on my phone.
Can you try that?
Which video would you like, Lisa?
Says the host has stopped my video, so I can't open my video.
Oh, okay. Let me open your video. There you go. You should be all set.
Thank you. Thank you.
You're welcome.
Greetings, everyone. Let's pull this up. So I was invited just in September to share on diversity, and I apologize. There is, I think, one replicant slide here and no intro slide. So we're in Belize. My name is Lisa Carne, Fragments of Hope. I am showing on the right-hand side of your screen the number of genets at 3 of our oldest outplant locations of staghorn. So at Laughing Bird Key, I'm in southern Belize where the white circles are. Silk Key and Moho Key, we have 17, 21, and 14 different staghorn genets outplanted respectively. The size of Laughing Bird is about 1 hectare or 10,000 meters squared. Moho is much larger and Silk Keys is much smaller. And Laughing Bird, we've been outplanting since 2010, well, 06, and then scaled up 2010. Moho and Silk since 2015. At each of these sites, we also have, now this is Palmata genets, 11 or 12 at Moho, at Laughing Bird 28, this is prior to this year's event, and 14 at Silks. So I wanted to share with you that, of course, you know what's going on with the bleaching. This is data from the Belize Fisheries Department showing again that, as you've all talked about, we had a big spike in October, even though we had an abnormally early spike in June. And we're still in it, as was mentioned. So it's abated, but it's not over. This is really busy. It's just showing you that for southern Belize, we have bleaching data going back to 08, and someone asked about the severity of this event. And it is the most severe that I've witnessed. I did not have a camera in 1998. So I can't speak to the event in 1998, which was global. But I did see a link with bad news about the El Nino forecast event, and they're saying that they're comparing this year to the same seriousness of 1999 and 1998, unfortunately. So this is the slide I had shared with you all before, that our strategy, as was mentioned, is to always map the surviving. And mapping this time of year is perfect to find out who's really surviving, and then including different genets so that they can eventually reproduce. And on the right-hand side was time series that I went ahead and updated. These were, from 10 years ago. And then I can't see my screen. So hopefully this is updated for July and then August. No, okay. Yeah, so there's more here. So then on the left-hand side, again, I think I repeated this before, this needs to be updated. This is just cervicornis, but we've been mapping all throughout Belize. Okay, there's October. So you're seeing the time series now. It's pretty dire, as you know. Then this is just a short video. Again, I had shared it, but now it has some updates of the same different genets outplanted at Laughingbird Key through October now. So you can see in the middle, one is living. So it's just a example again of why you need the different genets of each species, because there are going to be different patterns. This poor one has been bleaching since June, I mean, solidly. And then now you see mortality in the flat parts in October, but you do see some recovery. So there is variation. We have no idea what the difference is between the genetics of the host, the symbiont, or the coral rather, and the symbiont, and or where you place them in terms of depth, and or if there is any morphology, like Andrew was asking, you know, to differentiate. And so this is something we hope to explore in the future, using genetics and using both historical and future data to be collected. So again, this is just running through kind of the losing versus the winners. How do we talk about it? And hopefully I'll get to a point where I wanted to mention, if I don't say it later, that even some of these loser genets actually retain some tissue. And so I have some photos very recently at Moho Key from last Friday that's going to show you a gradation in all three of these oldest sites with the different genets. We have everything from completely dead to not bleached at all and everything in between. And so it's just a plethora of data to now go hunt down and figure out what's really going on. So these are sites at Laughingbird that never did bleach at all, really, I mean, barely compared to other little subsites at Laughingbird. And again, just running through the different reactions with, you can clearly see with your own eye to the bleaching with different staghorn, different prolifera, different palmata. I think that's enough of Laughingbird. Let me move on to the, this is now from Friday at Moho Key. And so this has no dates on it. They're all from last Friday at Moho Key. These were outplanted in 2015. I was trying to illustrate the different thickness of the branches. Here you see dead corals in the background. You see live corals. This is due to fireworm predation, where this, let me back up. Can I pause it? This is quite interesting if I can go back. Oh, maybe not. We have noticed a few of these genets here. I can't go back. Wait, yes. Sorry. Oh, I hit buttons too many times. Anyway, there's a few staghorn that can overgrow the fireworm predation. So on that picture, it was showing that like the new little branches coming out were bleaching. This is totally dead. Again, just illustrating all the extremes that the different genets are showing. Again, this is all the same date, last Friday at Moho Key, where we began outplanting in 2015. So from healthy and not bleached to everything in between. And now these are the palmatas at the same site from the same day last Friday. So some of these little guys are looking okay, some are not, right? And then we have to do all the background to say which genets are doing okay, where do they come from? That's going to be my next short video, is the donors. Here's the illustration of just a few little polyps hanging on. And again, these are the first first images were corals we just outplanted microfragments in 2023, and now I'm moving to like last year's, and then I'll end with the older ones. So these are from 2019. The 2019, I think, are the oldest set that we have of the microfragmented palmata outplants at Moho Key. And at this point, I'm happy to see even any tissue living on some of these, which is sad. So again, these are all from the same day last Friday, just swimming around the key to our different outplant sites. And here was two different genets. So you can see clearly the reactions between two different genets outplanted close to each other. Hopefully you can see the coloration difference. These are the same two genets here. When we first started, we were experimenting with putting them together. We don't recommend that anymore, but we wanted to see what would happen based on the genetic diversity that we had first looked at using Ileana Baum's methods on the natural palmata stands. And we had pretty high diversity, but now we're keeping them separate. So you're getting these individual colonies of different genets. This is only the staghorn, just to give you an example, and it's not updated, that we mapped all these donor, what we call the donor locations. And now then what we've started to do since September is revisit the donors. I'm showing mostly the elkhorn. This is an inner key site. What we mean by that, it's got higher temperatures and they're looking okay. This was also inner key, but not looking okay. This had been a beautiful palmata before this event. A proliferous stand that bleached in 2020, started bleaching again in June this year. Yet we've moved this genet around and it's actually doing better. Here it is at Moho last Friday, not bleaching at all. So the staghorn as well, we've seen the patterns of some of the donor reef sites where the staghorn came from are not doing as well as they are at our outplant sites. So that, I think, has to do with our rapidly changing conditions and that it's just gotten too hot too fast for them. So our lessons learned, I mean, we're still going through this event and it's quite overwhelming, is more diversity is always better. So far, regarding the palmata, I should rewrite that. The palmata donor corals seem to mostly be holding the patterns at the transplanted sites. And then we've been outplanting the staghorn mixed, and I still think that's a good idea. So we put as many as 2 to 5 different genets together on the same site. But I think moving forward, we want to keep the palmata within 1 meter or 2 of each other, the different genets. And finally, as was asked earlier, you don't want to remove bleached corals from nurseries until you're sure they're 100% dead. A couple examples I showed you where, you thought that coral was dead and these little polyps, maybe even 1 centimeter is hanging on. So for example, partners at a remote site for us in Placencia removed this bleach rope, and I was telling them, like, don't do that because there could be some little centimeter of polyps that could have recovered, but now it's removed, it's gone. And just to reiterate what Judy said, we do have in situ temperature, but I think this is graphic enough to show you that whilst we've had a lot of rain and temperatures are cooling and maybe we're seeing some recovery, we're not out of the clear yet. And as I mentioned, that El Nino prediction is pretty dire for the coming into 2024. And that's it.
Thank you, Lisa. And our first 2 speakers provided updates of their presentations that were given in September. We'll now hear for the first time about Jamaica in the Central North Caribbean with Felix Charnley of the White River Fish Sanctuary and the Resilient Coral Mapping Network. So take it away, Felix.
Thanks a lot, Judy, and thanks to the group for addressing Acropora in response to this crisis. So I'll quickly give you the lowdown then as quick as I can on the present scenario, what we're up against, what we're doing, how, and where we want to go from here. Y para los participantes hispanoparlantes me dispongo a contestar cualquier pregunta en español, por si voy muy rápido, cualquier cosa. So just to set the scene, I'll start with Andrew's data on the next slide from the St. James Montego Bay area. It's basically been hot since May. The peak from Andrew's loggers was late August. And then from mid-August, it kind of bubbled between 31.5 and 32. And then even in the night times, the reduction in temperature in early September was still over 30. And we think that was the final straw that sent everyone bleaching. We've basically been praying for a hurricane. The rough seas cooled the waters here and there through the summer, but all that came to a stop in September. Generally speaking, the decreased frequency in hurricanes and the increased temperature doesn't spell good things for corals because we no longer have that seasonal respite. And then the bubbling continued around 32 for our site here in Ochuris until at least October 15th. Then we started to see some rain, some waves, and it's been around 31 ever since. And we've actually seen some color return. For example, to our lonesome Dendrogyra that we actually found in the sanctuary the other day. And that also churned up the water somewhat. And that's reflected by the luminosity levels on the chart. So that probably helped a little bit too. Unfortunately, none of this can be said for Acropora. So I'm really just talking about the massive species. But on the next slide, as far as Acropora is concerned, we have not been dealing with a bleaching event for a long while now, aka since October. I wish, but for well over a month, it's been a mortality event that we have on our hands. Bleaching was then, extinction is now. So that's what we're attending to. This is a comparison over the course of 3 months at exactly the same spot on the drop-off on the western section of Rio Bueno. So you can see the 3 different stages of a mortality event. You've got the bleaching in the middle, and that's how quickly that can all take place. And then on the next slide, just so you can really state your risks, is you got to look at this buttress system of salt marsh in Trelawney, where you've got these expansive assemblages of all three acroporids side by side in abundance, completely wiped out, fresh, recent mortality. So this is basically the speed at which this local extirpation event has occurred for us. Acropora did not wait around this time, and any survivorship is exceptionally rare. So just eyeballing it, we haven't been doing transects, I'm afraid, because we've been so focused on rescue. But just eyeballing it, mortality is looking to have been over about 90% of everything that was there literally a month and a half ago, 2 months and a half ago. So our biomass is close to zero if we don't start propagating the survivors now, because most nurseries around the islands have also been wiped out. We still have some bleached staghorn in one of ours, but we need to check that tomorrow morning. Our gene pool is now incredibly small, and obviously our survivors are now more vulnerable than before due to isolation. Therefore, prompt rescue is in order. So on the next slide, we started off doing the shading thing. I think, we acted a little bit late. We shaded everything from one nursery plus some wild stuff. We started off with mixed results, but by now I can tell you straight up that the only stuff under the shades that's alive now is the wild stuff whose donors are still not bleached from the 8 to 10 that we have found to not be bleached. So that's not thanks to the shade per se, but we may have acted too late as bleaching had already set in, and they say you have to get them under the shade from before, but that wasn't my experience in 2020. So who knows? We can still do it as a mitigation in the future, but for now, it's all genetics from here on in, which is why we've been doing these epic swims across multiple parishes to find survivors, geo-reference them, flag them, and share that data on a digital map, photograph them, and periodically upload those photos to trace their evolution. So where you can see the pinpoints marking the colonies, I'm sorry, the image is so small at the bottom there. All the colonies that are there in total cover a 45-kilometer or 27-mile stretch as the crow flies. And that's not including the ones in St. James that Andrew and Yadira have found. So altogether, well, we've got the numbers. I'll share them on the next one. But it's been a concerted effort. So on a national level, WhatsApp has been incredibly useful for us to let one another know where we've been, who's mapped where we still need to go, if we can team up on certain dates. Our little group has got 20 participants in it now from 17 different parishes. And it's also really useful to have this network because there are people in the group who may not necessarily be reef managers, but they might live adjacent to sites where survivors are found. Therefore, they can provide ease of access to those sites, support us with the logistics, or monitor them of their own accord. So it's also important to remember that some of these sites will have very specific conditions, which can be the reason as to why the survivors are able to be there in the first place. So transporting them elsewhere could be inadvisable for the time being. So the best bet might be to set up an adjacent culture, which can be overseen by the community that lives right there, whether it's a hotel or a fishing village or whatever, until we can afford to start sharing material and indeed start working ex situ preferably as soon as possible. So who have we found? On the next slide, you've got some surviving elkhorn and staghorn, zero prolifera, which is a bit shocking, as over the years we kind of thought the hybrids would be somewhat tougher than the parents. The only good thing is at least the parents can produce viable prolifera larvae, I assume, when matched. But it would be very sad to see the loss of that one. But yeah, so we're looking at 5 cervicornis in that whole 27-mile stretch, 21 elkhorns, and then yeah, we've got that one pillar coral in the sanctuary. So as you can see on the top line, you've got the cervicornis, which is dwindling, largely fighting off competitive algae. On the right there, you've got some disease or some receding tissue, could still be the RTL. Those tips need salvaging before that's too late. And if that's all the cervicornis that there is for 27 miles, we need to be quick. We need to be quick, but we also need to be careful. In Palmata, there's a few of these, but for the most part, tiny colonies. I mean, so tiny that some are even just spat, like on the right. 1 or 2 medium-sized ones in the middle, and then the one on the left is kind of that's as big as they get for us. Happily to say that's actually very close to our base here at Noturius. And that's actually one of the donors of the 2 pieces that have survived under the shade cloth. So in terms of location, these are all at different depths. Sites tend to vary in condition, but generally speaking, any surviving material is surrounded by dead stuff. So we can't really put their survival down to site condition for the most part, but moving forward, say for in situ culture, we do want to take site into account, of course. So already mentioned how the survivors are up against predation and macroalgae, but some of the other threats include, so on the other slide, on the next slide, late bleaching, as in how long can they stay unbleached? So this oscillation has taken so long, who's to say 4 months in, they're still going to be okay. So we're just hoping that they don't, give in at this point. As for disease or not, question mark. I'd love to have a diagnosis for this stuff in the middle. We're keeping an eye on any progression. Again, we need to beat it, A, by salvaging good tissue for propagation while we still can and isolating that, and B, maybe sealing the lesion. I mean, we can't afford to lose these, so that's already half that colony gone, and that's one of the big ones. So, I'd like to have that discussion with the group. Maybe, we were using the amoxicillin-based B thing in Cayman with the DOE, specifically for Schedule D. On that scale, obviously, it's not cheap, but a solution like that for the less than 10 colonies that we want to treat, it could be an idea. I don't know what you think. And then the thing on the bottom is very similar, looking to the RTL, the thing that was being discussed in the last meeting. So from before the heat spike. And this on the next slide, you can see it on the bottom line. That's this stuff, that's the same RTL that we were seeing in August on the left. And then assuming that's the same thing, you got it on the right from like last week. And then on the top there, I'm kind of hoping that's just sediment smothering, but the tissue seems to be receding towards the tip, but one of Lisa's pictures was almost identical to that one there on the right. So, yeah, I don't know what's going on with that, but I'd like to. So I don't know how much time I've got left, but basically, where do we want to go from here? On the next slide, well, we basically start again from scratch. We do the mapping, we multiply the survivors. Maybe set up an adjacent culture to the sites where the survivors are found, because transporting them at this time may be inadvisable. I'm not sure. Temperatures may, still be too high, not ideal for propagation, or it could be that they're surviving at the sites for a reason. Could be depth, could be chemical composition, so we're not sure. But once we've got the numbers up, we can then start thinking or simultaneously start preparing, preferably to get samples of them into a land-based spawning facility where we can crossbreed the survivors from, say, White River with the ones from, say, Antigua Bay. Reintroduce the new recruits to the in-situ nurseries or strategic outplant sites where the genes can mix and match in the long term. Jamaica actually has a raceway at the Alligator Head Foundation in the Eastern Parish of Portland that can use, and we need to continue to work as an island-wide nation of practitioners to have a concerted national effort to match genetic material from far and wide. So this is where the WhatsApp group has been so good, but going forward, we want to keep that up because we want to generate that variation. And in the foreseeable future, maybe get, some of these guys like Coral Spawning Lab, because they're doing stuff in Cayman to maybe stimulate broadcast spawning. But we need to do it ASAP because the donors may not last too long in situ. But right now it's swim, 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 because if you're not going to prioritize the swimming, where are you going to get the corals from? So that's our number one. And that's pretty much what's happening with Acropora. I don't know if you wanted me to mention what might be happening with some of the Schedule D corals on the next slide, but I just briefly at this point, those species are the what's going to be left essentially for now. Like I said, we haven't done transects, but eyeballing it, I'd say the majority of those are still only bleached. Some of them even recovering. We're keeping an eye out on the boulders though. Judy was saying in our chat the other day how when zoos actually repopulate these, they can die quite soon after this, and we're not sure how that's related to white plague or Schedule D. So definitely keeping an eye out. But we have seen some Dendrogyra, which is great. And I know Andrew and Yadira have also seen some in the Montego Bay area. So.
Thank you very much, Felix, for that sobering introduction to the current reality in Jamaica. Our fourth and final speaker will be Emily Becker. Now, in September, we learned about the emergency responses that were being undertaken in the Florida Keys to move representatives of hundreds of acroporid corals from shallow to deep water. Simultaneously, and we didn't mention this, heroic efforts were underway to create ex situ nurseries on land, mostly for the SETLD susceptible corals. And Emily will now give us an example of one of these immense emergency efforts that was being done at the Keys Marine Lab. So hello, Emily, I see you're...
Good morning, everyone. Thank you, Judy, for that. Yeah, so I'm going to be coming at this talk a little bit differently from the land side of things. And so I'm just going to be going over kind of what we learned this year, what we're hoping to be able to do for next year. Fingers crossed it doesn't happen again, but just in case it does. And some of the struggles that our coral practitioners encountered during this bleaching event. So if we can go to the next slide, please. So just a quick overview of our facilities. We have land-based. We have 3 land-based facilities. Ooh.
What happened? What happened?
If I could ask everyone to turn and mute their audio and their video so we can have Emily talk.
Let me go back to the previous slide, please.
Sure. There you go.
Thank you. So we have 60 seawater tables on site. They range in size from 40 gallons to about 250 gallons. And we're lucky enough in that we pull water from a deep water well. And so we're not really at the mercy of bay water or oceanside temperatures. Our water usually comes in at about 24 to 25 C. So that really helped us out quite a bit. This water goes through degassing towers where it strips out some sulfide gases, and then it moves onto our tables where we sterilize, sand filter, all that kind of stuff before it reaches the table where the coral is going to be. We also have the ability to temperature control, manipulate. So traditionally, people are bringing their coral or other organisms to do research on. This year, all of our tables, 100% capacity, were used for coral rescue efforts and bleaching efforts. So we were trying to just maintain water at temperatures that we had seen in previous months in June and July and August, and not the 91 or 32 C that we were seeing in the offshore reefs. So on the next slide, this is kind of what happened for moving coral from offshore nurseries to land-based facilities. Unfortunately, we were not really prepared for this. The practitioners, coral practitioners down here, were starting to see bleaching at their different offshore nurseries, but they had to kind of just make a decision as to when they felt it was appropriate to start bringing coral in. It happened very quickly. We had quite a few groups that said, all of our coral looks fine, tissue's a nice dark color, and then 3 days later, they got back out to their nursery and almost everything was bleached. So it was kind of a very rapid, chaotic harvesting of these corals from offshore. We had about 5,000 corals come to shore, about 8 to 10 different species, endangered or threatened species. And you can see in this picture on the bottom, Coral Restoration Foundation, we're transporting a lot of their coral from the boat into coolers, into this, we're calling it the coral bus. And it was a temperature controlled trailer essentially to move stuff around. And not a lot of different groups were basically just throwing coral into coolers on the boats just to get them to land so that we could start acclimating them to the temperatures in our land-based facility. Next slide, please. So now we're just going to go through a couple of slides of just pictures of what the status of these coral were when they arrived on site. Bleached, pale, some having some tissue loss. And so a lot of them were brought to site in these coolers, in these crates, acclimated to temperatures on site, and then taking a sigh of relief and kind of figuring out what next steps were for long-term storage of these corals on site, right? There was no temperature drops in the offshore reefs in sight. And so figuring out how to keep them happy and healthy and hopefully recover in a land-based facility. Next slide, please. We did have a bit of a silver lining with this in that we saw that the boulder coral or the massives were actually responded pretty well to coming back to land-based facilities. You can still see that quite a few of them are bleached or pale or stressed in some capacity, but these tended to do a bit better in our land-based tables. Next slide, please. What was also encouraging is that a few diesel little fragments were brought to site and actually did really well. They were pale out on the reef and they made a full recovery once they were back on site for about a month. So that was very encouraging to see. On the top right picture, you can kind of see the different scale of bleaching for different acroporid species. We did have some MCAVs on site too, and a lot of OFAV on site, and they all tended to respond very well to being brought to cooler temperatures. And they were also shaded depending on what the different coral practitioners wanted to do. Next slide, please. This last one we'll show right here are some brain corals that were brought on site. Again, this is just to kind of show the different means that coral were brought to site and housed in the different tables. This did change over time as we had to reduce densities to help with water flow, and just overall, lower densities tended to encourage growth and recovery. Next slide, please. As I alluded to before, the acroporids were difficult to keep long-term on site. And so many of the coral practitioners who brought acroporids to site ended up hanging them in some type of tree formation. So it was a piece of monofilament wrapped around it. These coral tend to need quite a bit of water flow, water movement, not necessarily turnover, but just movement around them. These were extremely difficult for practitioners to keep alive long-term throughout the duration of this bleaching event. And so we learned quite a lot of lessons from growing these on site, and I'll talk about those a little bit more later on. Next slide, please. Another method, and again, this was something I alluded to before, we had to move from keeping coral short-term, which is what we were used to at KML, to keeping them more long-term. And so traditionally, a lot of coral were being just attached either for transportation or short-term duration in our tables to just these little trays or egg crate or whatnot. And it wasn't really working for a long-term strategy. And this was primarily due to the fact that a lot of the tissue was in contact with either the egg crate or plastics or PVC, and it was encouraging ciliate growth. And so that was something, again, a learning curve that we had to learn from. And coral practitioners really had to start figuring out what worked best for their particular fragments. Next slide, please. So just to touch on some husbandry that we gleaned from this event. In the top left picture, you'll see that there are a bunch of covers over the tables. The clear plastic in the middle prevents freshwater influx from rain. So these are outside facilities. It's single pass system, so flow through, not recirculating. So the water passes through the table one time. And there's also, you can see the black mesh over the tops of the tables. And so when coral came in and they were either stressed out, bleached, pale, we had the option of either 80%, 40%, or 20% shade cloth to put over those tables as the coral started to recover, and you could expose them to full par light levels. We also found that, I alluded to this before, we needed a lot of water movement. So those Voyager pumps were really beneficial to all of the acroporids, and just keeping water moving around, preventing ciliates from settling and causing any issues with recovery. Amino dips, reefroids, being able to actually feed and get nutrients into these corals as they recovered, as our facility, like I said before, is single pass, so not much is getting into the tables. It's very clean water. Being able to keep the tables clean, right? While we do have UV sterilization, UV filters, you're still going to get some algal growth. So having lithopoma available to kind of keep algae down and keep the workload off of the practitioners while they were keeping their coral on site. And lastly, I just want to touch on, we had a AZA heat team meeting. A bunch of people from a bunch of AZA zoos and aquariums across the country came and helped out the different practitioners with best treatments, husbandry, diagnostics, right? So many of these practitioners are moving from an offshore nursery to a land base, and it's really tricky. There's a really big learning curve there. Next page, please. So the last component of this event is everyone's asking, when can we get our corals back offshore? So NOAA and FWC, which is our local Florida Fish and Wildlife Commission, they put together a bunch of protocols and guidelines to make sure that diseased and stressed coral were not going back off the reef once water temperatures came down. And so they decided that after October 15th, that was kind of the cutoff date, and after the water temperatures were below 30.5 5C for at least a week, different groups could start making moves to move coral back offshore to their nurseries. So in preparation for all of this, the different groups needed to remove dead and diseased corals from their offshore nurseries in preparation for bringing the coral that were held in the land-based facilities. Additionally, a coral health veterinarian needed to come and do a full health assessment of all of the coral that they had been maintaining for the last few months and make sure that they were healthy enough to go out there. And then finally, all of this needed to go through the FWC Coral Restoration Protocol team to give them the final approval before they could start moving them back out. And so they have a short 30-day window to move these coral back offshore. So it was really thought out to make sure that stressed or diseased coral were not being moved from one facility to offshore and impacting potentially the wild populations. Last page, please. So take-home message from all this, what did we learn? There's a huge learning curve for coral practitioners moving from offshore to land-based nurseries. Acroporas were very challenging to hold on site, and as I talked about before, still learning a lot about that just for long-term instead of short-term. The boulders tended to be a bit more successful. Developing protocols, holding training sessions, and just teaching staff that will be doing the husbandry well beforehand so they're comfortable with everything. Like I said before, this was a mad dash. It was very stressful for everybody, and it took a long time to get everybody on the same page, and that was no one's fault. Biosecurity was another huge thing. While we are an outdoor facility, just really stressing biosecurity to all the staff on site. We had a lot of practitioners here, a lot of coral here. And then just learning to plan well before the coral need to be moved to a land-based facility. If that's the best option for you, you know, what parameters do you need to hit before you start pulling coral from offshore, how to transport them properly and effectively, and then again, any protocols, husbandry and such. So yeah, that was our experience this year. We're open to learning from anybody who's had maybe similar situations, and we look forward to chatting with you soon. Thank you.
Well, thank you ever so much, Emily. That was a huge undertaking, and you know, I really truly had not appreciated the fact that the nursery practitioners had to come in and help look after their own treasures during this whole time. Then of course there'd be no way on earth that the couple of you could possibly manage to take care of and supervise 5,000 corals bits.
That's quite an undertaking.
That had been brought in over a period of just what, a couple of weeks?
Yep, the early July.
Early July.
Yep.
So I hope you'll write this all up, because the lessons that you learned this year for any part of the Caribbean that has the ability to hold any vulnerable corals ex situ during an emergency like another bleaching event will surely profit enormously from what you have learned.
We are definitely working on it. It is in the works.
That's good to hear. And I guess we'll throw the floor open to questions for a little.
We have one question from Ruleo. If Ruleo, you'd like to go ahead and ask your question. And I'd like to ask all of the speakers who presented today to please turn on your video so that everyone can see you.
And I finally figured out how to turn the camera on this laptop.
Thank you, Emily. Really good talk. I do have a question though, because I know you spoke about that you guys, or you had corals that were brought in that were essentially pale or bleaching, and that you saw different levels of recovery. And then and you're saying you're pulling this water from a well. So I'm just curious about, were you doing anything to kind of encourage zooxanthellae into the system? Because I'm trying to understand how it is that if you have a closed system, or essentially a closed system, how are they recovering? How are they sourcing this additional zooxanthellae for them to recover from being a bleach state in that case?
Right. So that was a concern from quite a few people. And Dr. Cindy Lewis, actually, who is our director here, her PhD work looked at how many of these coral actually have their zooxanthellae from the get-go, and that once they reach adulthood, I guess you could say. You have to excuse me, I'm not a coral biologist. They can still make a recovery. They don't need the outside sources of it. And they're also all being, they're all in tables with other varying degrees of recovered coral. So there's the thought that maybe they can pull them from other corals as well. Yep. If anybody has better information about that, I'd definitely open the floor to them.
Thank you, Ruleo. Okay, do we have another question? If not, I'll go for some questions that were in the chat. Sarah from Inapesca in Mexico had a question for Esther. She said, during our Acropora monitoring actions in Puerto Morelos, we found pink spots on completely bleached apal, as shown in the pictures. Tissue was still alive and you could see the tentacles on the polyps, so maybe it was something coming from within. Have you seen anything like it, or do you have any idea what could be happening? Esther, could you unmute?
Yes, I looked at these briefly, and yeah, there could be something in the skeleton that has a pinkish pigment, and I don't know what it could be at the moment. We would, of course, need histopathology to help us understand this. So I'm sorry to say I can't say anything about it at this time.
Okay, thank you. Also, we had a question from Lisa to Emily. Was there a subset of corals left in situ to compare results with the cost and just how the corals did overall?
This differed from group to group, from coral practitioner to coral practitioner. So I know that quite a few groups did still have some coral out in the field, and they have just started returning some of their coral. So I think stay tuned for results from that. I think there will be a lot of information available over the next couple months.
Okay, thank you. There's also another question is, are the NOAA FWC bleaching husbandry protocols shared or accessible to other groups, or will they be eventually?
Yes, they did make them publicly available. I think you had to be part of a specific group for the first kind of send out of them. But I can check in and see if they are publicly available, and if we can get a copy and send them out to everybody. If someone's interested, leave your email address in the chat. I'd be happy to send it your way.
Okay, thank you. Do we have other questions from the group? I had a question for Felix, and it's really interesting how you incorporated both local community swimmers, and also you're looking to incorporate fishermen. You have a large coast there. Are there other areas that you're looking at? Because you were talking about how you're doing long swims and you're trying to identify. Maybe just touch on a little bit about how that's going to help you make your next steps for thinking about assisted fertilization or propagation.
Yeah, absolutely. It's a lot of ground to cover, and Andrew, feel free to chip in as well, because together we realized what we had to do, and that we needed to coordinate to cover all that ground and fill the gaps between us. But for example, we need people who have the stamina and commitment, and are long distance swimmers who will do like 5 kilometers per swim. So basically, Jamaica is divided into 14 parishes. White River Fish Sanctuary is located in Ocho in St. Anne, right in the middle of the North Coast. And between us, on either side of us, we have restoration programs. In the east, you've got the Acropora Bay Fish Sanctuary, and in the west, the closest program to us is Andrew and Seascape Caribbean in the area. And then in between that, there's Discovery Bay Marine Lab, but not an awful lot's going on there with the coral. Basically, we want to, from White River, to fill that gap. And we've almost finished. We need to close that up. So I need to do there's the section of Discovery Bay, Runaway Bay, St. Anne's Bay, Priory. Those are, and they have reef crest systems, so it's from either side of that. Yeah, so with the swimming, you've got to really zigzag because you're going to miss lots. You don't want to be on a boat, really. The boat can get you from A to B to your starting points, but you don't want to traverse this distance above the water. You have to be under. because you can't afford to miss bits unless you're going over, say, a sand patch or something. So yeah, we've actually tried. We've got 20 people in the WhatsApp group now, and we want to. I mean, I can't say how many of them are putting in the swimming miles, to be fair, but you know, we've got Rachel on the South Coast who's giving us information. And so, but we'd like to have people in Portland and St. Mary also, you know, but we can expand, and I've had help from a friend as well in Trelawney in the neighboring parish. So just teaming up, and then we can cover that ground. But we would like to, cover the whole island if possible. At least the group has representation from all parishes, almost. Well, half of them. And the reason that's important to doing any ex situ work is because we're like, well, for two reasons. We're not a rich country. We can't all afford to have a lab, but also we can't afford for the genotypes to be separate. We need to bring them all together. So that's also part of that. But we need the money to do that. But as a concerted effort, that's that. And then yes, in terms of fishermen, so actually, well, sorry, I go on forever about the.
Thank you, Felix. We do have another question from Marina. If you'd like to go ahead and ask your question, Marina.
Hi, thank you so much. Really, really great talk. I love this so far. I just want to say I'm learning a lot about diseases and effects of temperatures. I just put a chat, like my question in the chat, but I'll say it now. So I was curious about development in terms of, I've noticed a lot of like kind of complete absence of corals as if they died off a long time ago, and a lot of bleaching around the area where I currently am in Puerto Rico, and also around these very touristy areas of Mexico. And I'm wondering if anyone is studying construction runoff and the ways in which these very like quickly urbanizing areas are affecting, like basically if the pollution and the runoff from the massive amount of construction is affecting corals, and this would be unrelated to disease or rising temperatures. And like, is this something that somebody is factoring in? Or like, I guess my question is, and I'm sorry that it's unrelated to disease, but is this high, like development, pollution from development being factored into any of the ways that corals are dying?
Yes, probably. Maria.
Esther, do you want to go ahead and also answer?
Yeah, it is related to disease. That is the point I was trying to get across, is that all of these things are combining to more stress the corals, and that is going to be causing them to be weakened, unable to do what they can do. If the runoff is containing, for example, pesticides that can kill zooplankton, it's a problem. If it contains other compounds that may affect algae, that's going to either outgrow the corals or perhaps kill zooxanthellae. There's a lot going on here, and we just don't measure it because often the communities don't want to bother, or there's simply no money to do that. But it's very important to understand that yes, where there has been coastal construction, there are problems, and we see this along the east coast of Florida and all the Florida Keys as well.
Thank you, Esther. I think we have time for two quick more questions. We had a question for Lisa, which she's starting to answer in the chat, but I think it's an interesting question about, Lisa, are you trying to outplant corals deeper than the source colony? Is depth an important factor? Lisa, I didn't know if you wanted to expand a little bit on what you've been replying.
Sure. No, in the past, that was not our strategy at all. Our strategy has always been, until this year, to restore, replenish the top meter of living reef. So really focusing on reef crests and with the onus of shoreline protection in mind, right? But this year, I saw Andrew answer differently, but we did notice a significant difference in just a minute difference in depth. And again, I think it has to do with the UV light versus temperature, because unlike what other people reported, we see colonies that only bleached on top and not underneath. I know someone mentioned it's been a big debate this year about shading stuff and moving stuff, but I think for future plans, if we ever, I don't know when we'll outplant again, but if we ever begin outplanting again, even prior to this event, I had said for the palmata specifically that I was going to go just a little bit deeper because storms and natural fragmentation over time does move them more shallow. So if I can give them that little leg up for that first couple of years, and not much, I'm not talking about 40 feet, I'm just talking about a little bit deeper. But that's just our experience here so far.
Great, thank you, Lisa. I had a question for Emily. And the question was, Emily, you mentioned that it was difficult to keep acroporids in your tanks, especially if they were in contact with, whatever they were laying on, and you had to have more flow. And I was wondering, there was a really interesting paper by Nakamura and Robin Wosek back in 2001 and 2005, and I'll put the reference here. But the paper found, they were looking at Acropora digitifera from the Indo-Pacific, and they did studies where they used flow right above the tissue surface. It wasn't trying to mix the water column to reduce temperature. It was trying to go right along that boundary layer so that it could separate the boundary layer and reduce stress. And they found that it helped to reduce photoinhibition and that it helped to reduce the amount of stress. And I was wondering if we talk a lot about shading, but what you had mentioned about that actually needed more flow, are there opportunities to look at how flow just right across the boundary layer of the tissue surface could potentially help reduce the stress from either heat or irradiance?
Yeah, I mean, that's definitely an option. One thing we do have at our facility is that all of our structures are already 80% shade cloth covered. So that's already knocking down light levels to about 240 par, I believe. So yeah, individuals could definitely test that out, try it out. That's a great thought. I would love to look over that paper.
Okay, I put it here if you wanted to look over it, and
great.
Happy to talk to you more about it.
Thanks.
Okay, Judy, would you like to close us out because we are just a few moments over 1:30?
Yes, well, I would add to that if we're talking about temporarily bringing corals into ex situ situations in order to temporarily survive these horrid thermal events, we might also query the professional animal husbandry people in the aquarium industry, because they have figured out various techniques for keeping acroporas and similar highly sensitive corals alive. And that might be something to look into over the winter while we're waiting to see what's going to happen next summer. And thank you everyone who has spoken this morning, or has listened, or has asked questions, or has failed to ask a question because we didn't allow enough time. And if you're interested, we will post the chat, I think. Can't we do that as well, Patricia?
Yes, we can.
We can post the chat so that people can also see the answers that were given there, and look for the webinar recording in a few days, maybe later this week. So thank you very much for attending, and especially big thanks and hugs to everyone who presented. And good luck to all over winter. Let's hope the temperatures decrease for a while before we meet again.
And I just want to say our next CCT meeting will be in January, and we will get back together with everyone, and we'll send out an announcement to do more follow-up on these really critical issues.
Yes, including invasive alien organisms, I'm afraid. Bye.
Thanks, everyone.
Thank you. Bye-bye.